This is 11th class biology, chapter 13, photosynthesis in higher plants, and this is lecture number 1 of this chapter. Topics that we will discuss in today's lecture are introduction of photosynthesis and experiments related with photosynthesis. So let's start with photosynthesis. Photosynthesis is a physico-chemical process because both physical and chemical changes occur in photosynthesis, by which green plants use light energy, means sunlight energy, that can also be called solar energy, to synthesize organic compounds. Organic compounds, because glucose is made here by plants. Okay, so that glucose is an organic compound. It is the basis of life on earth because for all organisms photosynthesis is very essential. The plants give us the primary productivity. Okay, all other organisms are dependent on the productivity of plants, so it is the basis of life on earth. Ultimately, all living forms depend on sunlight for energy. Now, these are importance of photosynthesis. It is the primary source of all food on earth and it releases oxygen into atmosphere. Carbon dioxide is used in photosynthesis and oxygen is released in atmosphere. Now we will do about the experiments related with photosynthesis. First experiment is variegated leaf experiment. And variegated leaf is a leaf in which there are patches of yellow and green color. As you can see here, yellow and green areas are present here. So a starch test is done with this variegated leaf, and after starch test these yellow areas gives no color with on starch test. Okay, so areas unaffected by starch test means there is no color, while these green areas give blue-black color, as you can see here. So starch test is positive here for these green areas. It shows that for photosynthesis to occur, some green pigment is required. Okay, and also sunlight, because we done this in presence of sunlight. Okay, so sunlight and some green pigment is required for the photosynthesis. The same experiment can be done with a leaf, in which we can simple green leaf, we can cover half part of that leaf with a black paper, and after that we can do starch test. So only the uncovered part will show the positive test for starch. Second experiment is half leaf experiment, and for this experiment you can see this plant was taken and one leaf of this plant. So half part of this leaf was soaked into KOH solution. Okay, this flask was taken here and with the help of a cork it is completely closed, and in this KOH solution this half part of leaf was dipped. Okay, and KOH is mainly for the absorption of carbon dioxide. Okay, this KOH solution will absorb the carbon dioxide. It means for the half part of the leaf carbon dioxide is not available. After this this leaf was tested for the starch test was done, and only the exposed part of leaf was found positive for the starch. Okay, only in this part of leaf starch was present, and the part of leaf which was dipped inside the KOH solution, in that part of leaf there was no KOH, there was no starch formation, right? So we can do it by covering this leaf, half part of leaf, in a cotton soaked with KOH solution. Okay, so KOH soaked cotton can also be used here to cover the half part of leaf. Similarly, after that we will do the starch test and we will find the same results. So this is half leaf experiment, and it shows that carbon dioxide is required for the photosynthesis. Next experiment is experiments done by Joseph Priestley, and he performed a series of experiments in 1770 to show the essential role of air for the growth of green plants. And he done his experiment like this, like in starting he took a bell jar of this type and placed a burning candle and a mouse inside it, right? And he observed that soon this burning candle gets extinguished. Similarly, mouse would soon suffocate and die. Okay, so he concluded that burning candle or an animal that breathed the air that was initially present inside the bell jar, but only for some time, and because these are using the air that is present inside the bell jar, soon the air will be finished. Okay, and mouse suffocate and die. But when he placed a mint plant, so in next series of experiment, he placed a mint plant inside that bell jar along with burning candle and mouse, right? And now he found that mouse stayed alive, as you can see here. So mouse stayed alive and the candle continued to burn, right? So Priestley hypothesized. So this hypothesis was given by Priestley on the basis of this experiment that plants restored to the air whatever breathing animal and burning candle are using. Okay, so whatever oxygen is removed by burning candle, as you know, for burning also oxygen is required, and for breathing also, right? So whatever oxygen is removed by these, that is restored by the plant. It means plants are releasing oxygen, right? So it was hypothesized by Joseph Priestley. Next is about experiments by Jan Ingenhousz. So Jan Ingenhousz did his experiments from 1730 to 1799. Okay, and he conducted the same experiment by placing, same experiment that was done by Joseph Priestley. Okay, so he conducted the same experiment by placing it once in the dark, means the bell jar was once placed in dark and once in the sunlight. Okay, and then he showed that sunlight is essential to the plant for purifying the air, like we discussed in Joseph Priestley's experiment, that plants are essential for purifying the air. Okay, plants are responsible for purifying the air inside the bell jar, but these plants can purify the air inside the bell jar only in presence of sunlight. It means sunlight is essential for the purification of air by plants. Okay, so, and he repeated this experiment with an aquatic plant also. Okay, he took one aquatic plant also in place of mint plant, and it showed that in bright sunlight small bubbles were formed around green parts of the plant, while in the dark they did not. Okay, and later he identified that these bubbles to be of oxygen. Okay, oxygen bubbles were formed in bright light, while in case of dark these bubbles were not present around the green parts of the plant. And what are these bubbles? These bubbles were of oxygen. Thus he showed that only the green part of plants release oxygen in the presence of sunlight. Next experiment is experiments by Julius von Sachs in 1854. And by his experiments, he proved that glucose is produced when plants grow, and it is usually stored as starch. Chlorophyll is located in special bodies, that is called chloroplast, and glucose is made in green parts of plants only. So these things were proved by Julius von Sachs by different experiments. About those experiments, it is not in your syllabus. So you just need to do the conclusion given by Julius. Okay, that glucose is present in is made in green parts of the plant, and chlorophyll is located in chloroplast. Next is experiment done by T.W Engelmann in 1843 to 1909, and for his experiment he used a prism. Okay, and with the help of this prism he split light into its spectral components. As you can see here, these are different colors, different spectral components when light passes through the prism, right? Now he illuminated this cladophora, an algae. Okay, this is an algae. Okay, and inside this algae you can see these are chloroplasts, spiral chloroplasts. These are a spiral form you can see here. So he illuminated algae in this spectrum, and this algae was present in a suspension of bacteria. So all these bacteria are present in a suspension, and this algae is present in that. Now he found that these bacteria are able to sense the oxygen releasing areas, right? And you can see here these bacteria are accumulated here. These black dots are representing bacteria. Okay, so he found that bacteria were accumulated in two regions mainly. As you can see here, these are accumulated here and here. Okay, here in the blue region and here in the red region. So in these two regions especially, bacterial growth was found more. Okay, more bacteria were accumulated there. Why these bacteria are accumulated here? Because these are aerobic bacteria. These use oxygen for their growth, and these get accumulated in more oxygen releasing areas. Okay, now it was concluded by T.W Engelmann that this blue region of light and this red region of light, these two regions of light are responsible for the photosynthesis, or you can say for the release of oxygen by cladophora. Means more photosynthesis takes place in these two regions of light, in blue region and in red region. Okay, later on it was proved by many other experiments also. So you can say it was the first experiment about this, that how the blue region and red region of light are responsible for the increased rate of photosynthesis, because due to that increased rate only these two regions are responsible for more oxygen release and accumulation of bacteria in these two regions. Okay, it was proved by T.W Engelmann by this experiment. By the middle of 19th century, it is discovered that plants use light energy to make carbohydrates from carbon dioxide and water. And empirical equation of the process of photosynthesis is carbon dioxide, CO2 plus H2O. In presence of light, it makes C6H12O6, right? That is, you can write it like CH2O, N you can write here. Like you can write here N. Okay, so N is representing the number of carbon and these hydrogen and oxygen. Okay, like number of hydrogen will be double to that of number of carbon here. Okay, so it is C6H12O6 for the glucose, and oxygen is also released here, right? Where CH2ON represents a carbohydrate, that is glucose. Okay, so this is equation for photosynthesis. Now one more experiment is there which was done by Cornelius van Niel in 1897 to 1985. Okay, and Van Niel was a microbiologist. He conducted some studies in purple and green bacteria. He demonstrated that photosynthesis is a light dependent reaction, means it depends on, it uses the sunlight, in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. As you know that water is utilized in photosynthesis. Okay, so what is that water? That is hydrogen giving compound. Okay, So here it is writing hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrate. Okay, so plants use carbon dioxide from the environment, and they also use water. So from water these plants are getting hydrogen, and that hydrogen is responsible for reducing the carbon dioxide and to convert it into carbohydrate, means to glucose. So it was proved by Cornelius van Niel by performing experiments on purple and green bacteria. And he said that we can express the equation of photosynthesis like this, like it will be 2H2A. So we write for the equation of photosynthesis water here, okay, H2O, but it is H2A. H2A means A can be any substrate here, means any compound can be here which can provide hydrogen. Okay, in place of water any compound can be used here to give us hydrogen, right? Like in place of water, in case of these bacteria, purple and green sulfur bacteria, sulfur is used. So H2S is used here in place of H2O. Okay, and it combined with carbon dioxide which plants get from air, and in presence of sunlight it provides us 2A. What is A here? Again this S will be here. Okay, so it depends, it can be variable because all the plants use water for the photosynthesis, but these bacteria are using this H2S here. Okay, so this is any compound which is providing us hydrogen. Then CH2O, that is a carbohydrate, and water. So this was proved by Cornelius van Niel that we need a hydrogen giving compound to reduce the carbon dioxide to carbohydrate. He said that green plants, water is the in case of green plants, water is the hydrogen donor and is oxidized to oxygen. And in case of purple and green sulfur bacteria, H2S is used as hydrogen donor. So the oxidation product is sulfur or sulfate and not oxygen. Okay, so that we write 2A. What is that 2A there? That is S. Okay, that is S there, not oxygen. Oxygen is not released in those bacteria. What is released there? Sulfate or sulfur. Okay, thus he inferred that oxygen evolved by the green plant comes from water, not from carbon dioxide. Okay, so it was proved that the oxygen that is released, that oxygen is coming from water. It is not coming from carbon dioxide. What is happening with carbon dioxide? It converts into carbohydrate and water is releasing oxygen. And this later was proved by using radioisotopic technique also. Okay, later on it was proved by this technique. Therefore, overall correct equation for photosynthesis is this. 6 molecules of carbon dioxide we use and 12 molecules of water, and in presence of light these make 1 molecule of glucose C6H12O6 with release of 6 molecules of water and 6 molecules of oxygen. Okay, so this is the equation for photosynthesis. And this thing we were talking about, that water is releasing oxygen, not carbon dioxide. It was later on proved by radioisotopic technique. Radioisotopic is a technique in which we just make some of the like oxygen molecule can be made radioisotopic. Okay, and that can be visualized easily. In short, I am telling you that with making it radioisotopic, we can visualize it easily. Okay, so later on it was found that the same molecule of oxygen that we were using in this water, okay, the same molecule of oxygen we were getting here in the product. Homework for today is to read all the experiments related to photosynthesis from your NCERT book.